Hi guys, welcome back to my channel. I'm so glad to see you all here. So today's video is going to be about annotating variants and filtering them using GATK tools. So I'm going to continue the discussion from a previous video where we called variants using GATK best practice workflow, and I will make sure to add the link to that video in the description section below. So today we are just continuing that discussion, and we are going to talk about the next part after we have identified the variants. Next, we would want to ideally filter them and annotate them. So in the previous GATK video, we were following GATK best practice workflow to call germline variants. And in that video, we performed various steps which involve data pre-processing, starting with raw unmapped reads. We got analysis ready reads, and we use those reads to get, we call variants, basically align those reads and get variants. We use haplotype caller to call variants. And in this video, I want to continue using the variants that was called previously, and I want to filter to only retain the high quality variants, and followed by that I want to annotate these variants. So this video is more focused on what are the criteria behind filtering the variants to retain a high quality set of variants called, and what are the various strategies to filter variants, as well as annotating the variants, basically talking about what the annotation tells us about a particular variant. So once we have called the variants from a sample or from an individual, we may have some questions about the variant. So let's take an example. So in a previous video, we called variants from an individual that was a part of phase 3 of 1000 genome project. So now that we have called the variants from this individual, I may have questions like, if I'm interested in understanding or looking at a variant in a certain gene region or in certain genes, I may want to know whether this variant is occurring in the exonic region, in the intronic region, or in the regulatory region. I may also be interested to know whether the variant that is observed in that gene region, is it affecting the coding sequence after or is it affecting the translated amino acid sequence, and does it ultimately affect the protein function? Because if I'm trying to study a mechanism and the role of protein in a particular mechanism, then I may be interested to know whether there are any variants that is causing that is affecting the protein function. Or I may also want to know whether the variant that is occurring in that particular gene, is it commonly observed in certain diseases, or is it a benign variant that is commonly found in the genome? So there are various questions that one may have, and it's important for us to annotate the variants, because the annotations will basically give us some or maybe all this information about the location of the variant, the effects of the variant, and whether these variants are also present in other data sources, which I'll be talking about in the upcoming slides. So one of the questions that we were hoping to answer once we've identified the variant is that, is the variant occurring, is it a commonly occurring variant in a human genome, or is it a benign variant, or is it a disease causing variant? Is it observed in specific cancers, or is it associated with a specific phenotype? So there are specific databases or data sources that one can use while annotating the variants, which can give us which can help us answer some of these questions. So some of the commonly used data sources are Nomad, DBSNP, ClinVar, and Cosmic, if we are dealing with if we are trying to study cancer mutations. So just to quickly go over these data sources, Nomad, which stands for Genome Aggregation Database, it's apparently a largest collection of variation across the population. So, and it can be used to identify whether the variant that we are seeing in a particular gene or a region, is it present in every genome, or is it present in a specific collection of a population, or is it a pathogenic variant? We can also look at DBSNPs, which is apparently a database that contains all the identified genetic variation and can be used to investigate a wide variety of genetically based phenomena. We can also use a data source like ClinVar, which contains variations observed in individual with or without phenotype. So it supports the clinical validity of variance that is observed. And lastly, we also have Cosmic, which is commonly used when we are dealing with somatic mutations, or we want to study mutations that are linked with cancer, because this database contains manually curated variants relating to cancers. So depending on the type of question that you are asking, or whether you want to try and find out whether this variant is linked to a cancer or linked to a particular phenotype, or is it a commonly occurring variant? You can use one or multiple of these data sources while annotating your variants. And I will demonstrate how we use some of these data sources when we are annotating the variants using Functotator from GATK. So before we jump on to annotating the variants, we first want to filter the variants, and the goal of filtering step is to retain high confidence variant calls. So as we all know that the variance in a VCF file has various statistics associated with it. So when we were previously discussing a variant calling file format and we were understanding how to read a VCF record, we discussed some of these statistics. And if you're interested to know and learn more about how to read a VCF record, then I will make sure to add the link to that video in the description section below. So as we know that there are various statistics associated with a variant. So here, for example, in this figure, we have a SNP that is present at chromosome 22. And there are statistics like associated with this variant, like number of chromosomes carrying the alternate allele. We have information on the total number of chromosomes. We have allele frequency, depth of coverage, so on and so forth. So in order to filter variants and only retain high confidence variants, there are two approaches basically. So the first method of filtering is the basic traditional way of filtering variants, is to look at the statistics associated with each variant and coming up with certain set thresholds, and essentially keeping only those variants that fall within the thresholds and filter out variants that fall either above or below the set thresholds. The problem with this approach is that you can end up throwing good quality variants because one of their annotations look bad, or you can keep bad variants if in order to keep the good variants when you're trying to when you're trying to keep your thresholds not so stringent. The other alternative is to use a more sophisticated method, which is variant quality score recalibration method, which is essentially a machine learning algorithm which tries to learn from each dataset what is the annotation profile of good quality variants versus the bad quality variants. However, the downside of using this approach is that it requires well curated training resources, because since it's a machine learning algorithm, it requires good curated and quality training data from which it learns the annotation profile of good quality variants versus the bad. And these well curated training resources are not always available for organisms other than humans. And it also requires large amount of variation sites to operate properly. So it is not possible, or it's not suitable to use this method when you have small scale experiments or such as targeted gene panels, or fewer exomes, like less than 30 exomes. So workaround for that is to pad your cohort with data like exome data or genome data from other studies like 1000 Genome Projects. However, this workaround is not possible for data or variants called from non-human organisms. So for the purpose of this demonstration, since I've called variants from one individual, I'm going to use a manual method, a hard filtering approach, where I'm going to set thresholds for certain statistics to filter variants to retain only high quality variants from called from one individual. And followed by that, I'm going to annotate these variants. The filters that I will be using today will be applied at site level as well as at the sample level. So here we have an example VCF file, and we have previously discussed the data or information stored in each of these columns in detail in our VCF file format video. So the filters that I will be applying for at the site level are these. So basically these are the recommended thresholds by GATK that I will be using for both the SNPs and the INDELS. I will be adding the link to the GATK document in the description section below that explains what each of these statistic or metric mean. Now talking about the sample level filters, I'm going to use two filters. I'm going to filter by depth as well as by genotype quality. So essentially I'm going to filter out all the variants that have filtered depth less than 10. I've set the threshold as 10 because the data that I'm working with has low coverage, but I recommend you to set this threshold and tailor these thresholds according to your study design and the kind of coverage you have for your data. Next, we're going to filter the variance by genotype quality. So basically, genotype quality tells us how confident we are that the genotype we assign that is assigned to a particular sample is correct. So it's basically a Phred scale probability score. So to give you an idea, a genotype quality of 3 means that there is 50% chance that the call is incorrect, and a genotype quality of 20 means that there is 1% chance that the call is incorrect. So basically, I'm going to filter out all the variants that have genotype quality score of less than 20. So lower the score means higher is the chance that the call is incorrect. So an optional step here before we annotate the variants is performing genotype refinement, and the goal of this step is to use additional data to improve the accuracy of genotype calls and filter calls that are not reliable for downstream analysis. This additional workflow, like variant calling workflow step, is intended for researchers whose work is highly reliant on identification of individual genotypes, and this workflow is more suitable for analysis that are concerned with how many copies of each variant an individual has, or with the transmission of a variant in a family. So the data that we will be using for today's demonstration is what we used for the previous GATK workflow video, where we called the variants. So we are going to use the variants that is called from the previous video using haplotype caller. Again, as I previously mentioned, I will be adding the link to that video in the description section below. So in this video, we are going to use the variants that are called in the previous video, and we are going to use those variants to filter and annotate them. The prerequisites to follow along with me in this demonstration remains the same. I will not be going to the details of explaining the basics of Linux systems, or I will not be going to the details of how to write bash scripts, understanding Linux file systems and permissions, and I will not be demonstrating how to download and install command line tools. So I will just be requiring GATK 4 for today's demonstration, because I'm going to use all the GATK tools and functions to filter and annotate the variants. So before we start writing the script, let me show you the folder contents. So as I said that we are continuing using the variants that were called in the previous GATK video. So this was our folder structure, and our variants that were called are in the results directory. So I'm just going to show you that we have raw INDELS and raw SNPs, which we selected out of raw variants that were called using haplotype caller. So let us go back to our script now. So here at the beginning of the script, we have set the paths to the directories for the reference file and the results folder in a variable. We have previously done this just to keep our script a little more cleaner and concise. So the first step here is to filter variants, and we are going to be using hard filtering. We are going to set thresholds to filter SNPs and INDELS, and these are the thresholds that are recommended by GATK 4. So we are going to use GATK's variant filtration function to filter variants. We need to provide it with a reference file, and the reference is stored in the reference variable here. So we're just going to provide the variable name. Followed by that, we need to provide the raw SNPs, which is stored in the results folder, and the path to the results folder is stored in results variable. Followed by that, I'm just going to go to the terminal, and within the terminal we have the files saved in results. So the file where we have raw SNPs are stored in a file called raw_snips.bcf. So I'm just going to copy the name of the file here and paste it here. Followed by that, we need to also provide the path to the output and the output file name. So I want the output filtered SNPs to be stored in the results folder itself, and instead of raw SNPs, I want the filtered SNPs to be stored in a file called filtered_snips.bcf. Once we have mentioned these parameters, we will now start providing the filters. So we first provide a filter name for each filter, and then we provide the filter. Again, I'm following these filtering recommendations from GATK. So these are all the filters that I want to apply at the site level, and these are the recommendations by GATK. But now I also want to apply filters on the sample level at the genotype, and for that I will use parameters genotype filter expression and genotype filter name. So again, the format is the same, that you need to provide the filter expression as well as the filter name. So we are going to filter by depth, and the filter depth as well as the genotype quality. So let's write in genotype filter expression. For the INDELS, we are going to be using some of the similar filters. So I'm just going to copy and paste the previous block of code and remove the filters that we are not going to use. And I'm just going to change certain thresholds for INDELS. And also let us change the name of the input and the output files. So the input file was raw_indels, and I want the output file to be filtered_indels. So now let's save this. Let's go back to the terminal and let us run the script. It's just finished running. So let us go back to our results folder and see that now we have, in addition to the raw SNPs and indels, we also have four additional files: filtered indels, filtered SNPs, and the indexes for each of these VCF. So let us take a look at one of these filtered SNPs. So when we open this file, the first few lines will tell us about the filters that were used and the description of each of the filter. And scrolling down to the records section in the VCF file, here we can see that under the filter column, you will be able to see the name of the filters where the variants have failed that filter. So you'll be able to see that all these variants have filled the mapping quality filter that was applied. In addition, you will also be able to see that in the format column, which is this column, you'll be able to see that there is an additional field added. So all the genotype filters that failed, like for example, all the variants that failed the depth or the genotype quality, would have an additional field called FT with the name of the filter. So this filter failed the depth filter. This variant essentially failed the depth filter. If the variant did not fail the genotype filter, then you will not be seeing this field present in the format column. And if I scroll further down, you will also see that the variants that did not fail any filters have a value of pass in the filter column. So in order to select only those variants that passed all the filters, both the site level filters as well as the sample level or genotype filters, let us go back to our script and select the variants that passed the filters. So again, we are going to use the GATK select variants function, and we are going to use a parameter called exclude filtered. We are going to provide a VCF file. So the VCF file would be the filtered SNPs and the indels. So I'm just going to copy the command from here. So the VCF file would be the path to the filtered underscore SNPs .vcf and the file name. And I want the output again to be in the results folder, but I want it to be called analysis ready SNPs .vcf. And I will be repeating the same for the indels as well. And let us save this, go back to the terminal and go to the scripts folder and let us run the script. Okay, so now this is finished running. Let us go back to our results folder and Now we can see that we have another four VCFs, another four files rather generated, which are analysis ready INDELS and SNPs. Those are the VCFs, and we also have the indexes for those VCF. So let us take a look at analysis ready SNPs first. So when I scroll to the records section, I can see that now I have retained all the variants that have the values passed in the filter column, meaning that these have passed the site level filters that we have applied. But if you notice that it did not filter the filters that we applied at the sample level, at the genotype level, so you will still see that there are a lot of variants having the FT field present in the format column, and we can also see the filter, why the variant failed, what were the filters applied, why did it fail. So it failed the depth and the genotype quality both, for example in the first variant. So we also need to get rid of these variants that failed the filters that were applied at the genotype filters at the sample level. So for that we need to perform some manipulations. So I'm going to grep only those fields, or rather those variants that have passed both the genotype as well as the site level filters. So basically what I'm going to do is I'm only going to grep those lines that do not have the DP filter or the GQ filter strings that are present in the format column. So basically I'm just going to pattern match. I'm going to inverse pattern match. Grep is a very basic Linux command where we use it to match a particular string or a pattern and retrieve only those lines that match the pattern. But there is also a way to inverse grep to retrieve those lines that did not match with the pattern or the string that we provide. And let me show you how we do that. So I'm going to inverse grep. I'm going to provide multiple strings to match. So the first is going to be DP filter, and the second is going to be GQ filter. So basically this command is going to look for all the lines that does not have either DP filter or GQ filter or both. So basically these will be the variants or the lines that have passed both the site level filtration as well as genotype filters. So when I'm just going to do a less so that we can see the output before we save it into a file. So now that you can see that we have retained all the variants that have a pass value in the filter column, as well as we do not have that FT field, meaning that these variants did not fail the genotype filters that were applied. And you can also confirm that by looking at the depth and the genotype quality values for each of the variants. So now that we are assured that we have removed all the variants that have failed filters, now let us save these the output of the filtration to another file. So again it's going to be analysis ready SNPs filter GT, so that I can recognize that this is the file that is filtered by the genotype filters, and I would be using this the variants that are present in this file to further annotate. So I would be running this, followed we would be repeating the same for the INDELS as well. I'm just going to copy and paste from above, and let me also paste these commands in our script, just so that we remember that we use these commands to exclude the variants that fail the genotype filters. So going back to the script, I'll just make a note of these commands. Okay, and I'm going to save the script. So now that we have excluded all the variants that failed the filters, the next part of the video is to annotate the variants, and we are going to use a tool called Functotator from GATK to annotate these variants. So Functotator is an annotation tool by GATK, and this is the Functotator information tutorial page by GATK, and they provide extensive documentation on how to set up Functotator to run with specific data sources, and what are some of the pre-packaged data sources that are available with Functotator. They've also provided information on how to set up config files if there is a specific data source that is not included in Functotator that you want to run and annotate your data with. So today, for today's demonstration, I'm going to use the pre-packaged data sources that come with Functotator, and that can be downloaded using these commands. So since we have been following the GATK best practice germline workflow, I'm going to use the germline data sources, and these data sources have already been downloaded. But if you're running this for the first time, then you should be downloading the germline data sources on your machine before you try and annotate your data. The other thing that I want to talk about with Functotator is that one of the pre-packaged data sources involves Nomad, but Nomad is disabled by default. So if you want to annotate your data using Nomad, then you will have to untar the Nomad files. And again, Functotator becomes a little slower depending on the machine's internet connection, because once Nomad is enabled, the Functotator queries the internet when Nomad is enabled, and that can impact the performance. So if you do not require annotations from Nomad, then I would recommend to not untar these files and to run with the pre-packaged data sources. But if there are any specific data sources that you want to annotate your data with, then there is all the documentation information you require here to set up the config file and to have the data source in your file system. And they've also specified the directory structure, how you have to get your data, the data sources that you want to use, how you need to organize your data for Functotator to be able to access these files and annotate your data using the specific data source that you're looking to annotate with. So let me show you where I have downloaded the Functotator pre-packaged data sources on my system and how that looks like. So I'm going to go to the folder where I have Functotator downloaded. So essentially, when you run the command to download the pre-packaged data sources, you will essentially be downloading a tar.gz file. Once you untar this, you will be able to untar a folder. So this is the Functotator folder, and when I go to this folder, you will be able to see that there are some data sources that are already present. So as I mentioned, you will be able to see Nomad tar files, and I want to use Nomad annotations when I'm annotating my variant. I want to use Nomad as a data source, and hence I have untarred the Nomad exome and the genome tar files. In addition to Nomad, you'll be able to see ClinVar and GenCode as well. So Functotator runs GenCode, uses GenCode as a data sources by default, but you'll also be able to see it also use ClinVar as well as Nomad to annotate the variants. So when we're trying to run Functotator, we will be essentially providing the path to this folder as a parameter in the Functotator function, and I'll show you in a minute how we set up the Functotator command. So we will be providing the path to this folder so that it is able to look for the prepackaged data sources and annotate the variants using the data sources that is present in this folder. So let us go back to our script now. So to annotate using Functotator, we use a function called Functotator from GATK. The first parameter is the variant, where we provide the variant file that we need to annotate. So essentially we start with SNPs first. So the analysis ready SNPs, which were filtered for both the site level and the genotype filters, are stored in a file called analysis ready slips filtered gt. So I'm going to provide that as the value for the variant parameter. Followed by that, we provide the path to reference, and that's stored in the variable called ref. Followed by that, we need to provide the reference version, and we're using HG38. Next, we need to provide the data sources path, the path where our data sources are present. So that is what I just showed you a couple of minutes ago. So this is essentially the path that I will be using. Next, we need to specify the output. The output will be in the results folder again, and I'm just going to add Functotator, so I will be able to distinguish the annotated variants from the unannotated ones. And we need to specify output file format, which is going to be VCF. So now we need to do this again for INDELS. So I'm just going to change some file names. The rest remains the same. The input and the output file names will change. The rest should remain same. And I'm commenting out the code previously because I don't want to rerun the whole script again. I just want to run the last 2 commands. So now that this looks good, I'm going to go to my terminal and run the script. Now that the Functotator is finished running, let us take a look at the output files. So we have 2 annotated VCFs generated and the respective indexes for INDELS and for SNPs. So let us take a look at the annotated SNPs file first. So while I'm scrolling this file, at some point in the header section in one of the info tags, you will be able to see the Functotation fields that are present, the data for the Functotation fields that are present in this VCF file. So if a variant is found in any of these data sources, then the columns or the fields corresponding to the data sources will be populated. Now going further down into the records, we'll be able to see that in the info section, under the info column, along with the fields that we have an additional field called Funcotation, and within that field, the values of that field are the annotations. So right now, looking at these annotations, it is very hard to say and decipher the values that are present in one or multiple of those columns. So it will be helpful to have these values as in a form of a table, so that we can take a look at the variants and the annotations that are present in one or maybe multiple genes of interest, and maybe we can then try to create a plot or a figure, or try to understand the variants and the annotations that are present in our genes of interest. So let us go back to our script. So now we can extract certain fields, information from certain fields in the info column, and by those fields I mean specifically these values. So basically I can get these values in table. So basically GATK has a function called variance to table that will extract values from these fields and put it in a form of a table that will help us to understand and evaluate these metrics and try and understand the annotations that are present in each of these fields. So the first thing we provide is a variant file. So we are going to use the annotated script that was generated in the previous step. Followed by that, we specify the fields from where we want to extract the data that is present in the info column. And finally, we provide an output file. So I want the output to be written to the results folder, and I want the name of the file to be output snips.table. Now we save the script. Let's go to the terminal and let us run the script now. So now this is finished running. Let us go back to our results folder and let's take a look at whether the file has been created. The file has been created, and now let's open this file to take a look at the folder, the file content. So now we can see that some of the data has been fetched into columns, but again the problem with the Funcotation column is that the entire field has been lifted, and the value of the Funcotation column has been the entire field without like separating each of these values into different folders, because as we know that each of these value belongs to certain fields that we saw in the header section of the VCF. So we further want to separate out these values into a table, because essentially these are the annotations for the variants which we are interested in looking at. Now to extract these fields into a specific in a table format, basically to have these values into separate columns, we need to perform some data manipulation steps. So before we extract these values and manipulate them to save these values into separate columns, we first need to know what each of these value mean, what are the column headers or the column titles for each of these values. And if you recall, we did see that in a header section of our VCF file. So I'm just opening the annotated snip file again, and you will be able to see that these are essentially the fields. So the first field is the Hugo symbol, the second field is the build, the third is the chromosomes, so on and so forth. So I want each of these fields to be the column name, so that I will be able to understand the values that are stored in each of these columns. So there are two things I want to do here. First, I want to extract this line, and the second thing I want to do is I want to replace this pipe operator with a tab character, so that when I save this into a file and open it in Excel, I'll be able to see each of these values into separate fields. So let me show you how we do that. So this line has something like Funcotation fields are. So I want to extract this line. So let us grep this line, and now let me show you the output of that. So out of the entire file, we are able to extract this line. So now I'm just going to use some operation to replace these pipe operators with the tab character. So for that, I'm going to use sed command, and I'm going to substitute pipe operator with tab, and quickly showing it to you now that you can see that the pipe operators are gone and each of the field has been separated using tab. So now let's save the output to a file called output curated variance.txt. So now that we've extracted the column headers, now we need to extract these values basically. Now we need to extract these values. And again, we will be doing the same thing. We will be extracting these the values that are stored in this column, and then replacing the pipe operator with the tab operator. So I'm just going to cut the field. So this field is the fourth field, I think, or the fifth. 1 2 3 4 fifth field. So I'm just going to extract the values from the fifth field. So I can cut the fifth field, and I'm going to grep only for a gene of interest. So my gene of interest today is NBPF1. So this is the gene that I'm interested. I'm only interested in looking at annotations in this gene. So I'm just extracting this gene. Followed by that, okay, before we do that, let us just take a look at the output. So we have been able to extract the annotations that was present in the Funcotation column, that was column 5th. And under that column, we were only interested in few rows, the rows that were corresponding to NBPF1 gene. So I'm just interested in that gene. So we are extracting only those rows. And followed by that, we are going to apply the same set function to substitute. So we are going to substitute the pipe operator with the tab. And now let's quickly take a look. So now we can see that the values have been separated. Again, it's hard for us to make sense of this right now. So we are just going to save the output to the same folder. But now we are just going to append to the output curated file, the file that we previously created with the column headers. So I'm going to append the output to that file again. So now I'm going to open this file in Excel, and I'm going to show you all these operations that we have done. It will finally make sense once I show you how this file looks like. So when we open this file in Excel, you will be able to see that now you have information much more curated and in a form of a table. So essentially we have retrieved the variants that are found in this gene, and this is the chromosomal location. We will also be able to find the location of where this variant is occurring in terms of the in terms of the gene, in terms of whether it's occurring in the intron or in the 5' or 3' regions or in the exons. What kind of variant is it? We also get information on the genome change, in terms of the change in the coding DNA sequences. So these are all the gen code annotations. So since we also use ClinVar, you will be able to see that we do not have any information populated under the ClinVar columns, because apparently these variants did not match to any reported variant in the ClinVar database. So these are largely empty. And while I'm scrolling, we also use nomad. So you can see that there are certain columns in the nomad that are populated, and these are allele frequencies and various populations that are reported. So for these variants that have been present in the nomad, you will be able to find the relevant information from the nomad being populated in the nomad columns. So having these annotations and having them in this format is helpful, because this can easily be imported in R or Python, and plots can be generated. So if you have annotations for multiple genes of interest, you can create essentially plots of what kinds of variants are present in these genes, and what are the genomic changes or the changes in the coding DNA sequences that are found in these genes, as well as allele frequencies from the nomad, or if there are any ClinVar annotations that you want to visualize for your genes of interest. This can be done using the data that is annotated, as well as having the data in this format. So that brings me to the end of this video. So that's how we basically filter and annotate variants. I used GATK recommendations for filtering mainly, and I used Functotator, which is a GATK tool, to annotate variants. I will also be adding the links to all the references that I made during the video in the description section below, as well as upload my code to the GitHub repository, of which you will be finding the link to my GitHub repository in the description section below as well. If you found this video helpful and informative, please make sure you hit the subscribe button, like the video, share it, and leave your comments under the comment section. Until next time, see you.